So thank you very much, FDA, for putting out that guidance, even though it's draft, even though we are still waiting for comments. There is a guidance for the industry. So for anybody who is questioning or who is asking themselves, can we use those whole slide images? Like, how do we use them? It's written right here.
Learn about the newest digital pathology trends in science and industry, meet the most interesting people in the niche, and gain insights relevant to your own projects. Here is where pathology meets computer science. You are listening to the Digital Pathology Podcast with your host, Dr. Alexandra Zhurov.
Welcome, my digital pathology trailblazers. I can't believe it's the end of the year. It's actually 10 days before Christmas Eve when I'm recording this. I hope I can get it out to you before Christmas. So let's just take this moment and wish ourselves Merry Christmas or Happy Holidays, depending on what you celebrate. But I wish you and your loved ones beautiful, beautiful Christmas/holiday time so that we get some downtime and we can recharge for next year, because next year is gonna meet us with new challenges and we have to be tough. So today, actually, the topic is not very Christmassy. It's about an FDA guidance, but I love this guidance and you will know why in a second. This was a live stream I did on LinkedIn. And by the way, if we're not connected on LinkedIn yet, be sure to connect with me. I would love to have a conversation with you there. So this was a LinkedIn/YouTube live stream, and I was going through the FDA guidance for use of digital pathology in the preclinical GLP work. So this is for all my ToxPath people, but also for non-ToxPath people so that you know what the ToxPath people are dealing with. So have a listen. So today we're going to be talking about what FDA is saying about the use of whole slide images for toxicological pathology. So I have my guidance document here with me. Basically, I'm going to tell you what. So this is guidance for the industry. Let's have this one highlight, guidance for the industry. So what's happening here is we have a document. They're saying kind of what is recommended. It's a draft guidance as well, because there was a comment period where people were able to post comments. But they're saying it's just a guidance. No, don't take it too literally. Guidance is structured in a very nice way. I'm going to show you. But who am I even to talk about this? Is some guidance. I am qualified, or I self-qualified myself to talk about it, because I read this document 5 times or probably maybe 7 times, analyzing this for different things that I do for my work. And I have also presented it internally at several occasions. So I decided to present it to you as well, so that you don't have to go and read it, because honestly, it's not the most thrilling document. I mean, it's FDA document, all due respect. The goal of this document is not to be entertaining. So I hope I'm going to be a little bit more entertaining than just reading this document. So guidance for the industry. Our guidance has an introduction, background, and questions and answers. What we're going to focus on are our questions and answers. Why is it not doing anything? Anyway, questions and answers is going to be what we're going to be talking about. Okay, I can highlight. Fantastic. Before I talk about those questions and answers, let me know, were you familiar for this? Were you familiar with the fact that FDA actually said something about, oh, this is how we recommend you use digital pathology for non-clinical pathology, for toxicologic pathology. Toxicologic pathology was everything that happens before the drugs, before the compounds are actually administered to humans. So this is not the classical primary diagnosis thing where we have medical devices. It is more GLP, it is GLP, good laboratory practice, regulations. This is Code of Federal Regulations. But let me know, did you know, yes or no, that this is out there? And you can download it at any time. Question 1, what is whole slide imaging? Whole slide imaging, this question probably we can do it very fast because everybody interested in digital pathology knows what whole slide imaging is. This is this digitization of glass slides. So here, generating two-dimensional digital image, and here is an interesting part here. Two-dimensional. We are not talking about anything three-dimensional. We're not really talking about something that is dynamically zoomable, and this is two-dimensional. So with just a flat image. A glass of histology slide used for routine assessment in generation of pathology report. So the data in the non-clinical world, in the preclinical tox path world, whichever word you want to use, is the pathology report. This is considered data as per GLP regulations. So if we are using whole slide images for this particular purpose, we are this is what we are generating our data off of. So yeah, and we can see here that due to some limitations of the scanning, FDA does not consider the resulting digital image to be an exact copy of the glass slide. It's not an exact copy, but it is. They're basically telling us here it is good enough. There is a term called faithful replica, that it is a faithful replica. It's good enough to generate data on, to generate pathology report on. And then the question comes, should whole slide images be retained? And the quick answer, yes, it should be retained. So what's happening here is, if whole slide images, in the previous question, we saw, okay, whole slide images are good enough to do your job as a pathologist on, if you're a toxicologic pathologist, if you're doing toxicological evaluation. Good, one green light. Second question, should you retain them? Well, if you're doing your work on glass slides, then you retain them. If you are now doing your work on digital slides, logically you retain them as well. So GLP, if let me know if you are working in a GLP environment. Put a GLP if you are, and if you're not, put an NGLP, non-GLP. Because for those who are in GLP, that's going to be obvious, but I want everybody to, take it home. So for GLP, there's a lot, the main focus, or like the focus, is on traceability of your data, on being able to retrieve and trace the information and retrieve why you got to the results. So if there ever would be something, I see here we have some GOP, fantastic. But yeah, so in the previous, and yes, I will post the link to the guidance. I'm going to post it in the description of this broadcast. So totally, it's going to be there. So, but long story short, you retained glass. Now you're doing your job on digital images. Yes, you retain digital images. And what do they tell about this? They say that if whole slide images are assessed in lieu of original glass slides, so if you're now doing your job on digital, then yes, you have to retain it. So here is a little caveat, because it's during histopathology assessment and or pathology peer review. So currently, what is peer review? Peer review is a review, like I'm reading a toxicologic study, a toxicopathologic study, and there is a peer review as part of this whole study. Peer review is that somebody else is looking if I did my job well. Basically a second pair of pathology eyes, and this is part of the GLP study evaluation. it's a process that's happening routinely. It's not that, let's say, oh, I only have so many years of experience. There's always, this is very often, not always, but very often included in the study design, that there is going to be a peer review. Somebody else, usually from the organization that is commissioning this study, is going to check, okay, do they agree with my diagnosis? They usually have more information about the compound I'm evaluating, about previous studies that has been with the compound. So they want to see, okay, am I seeing stuff that's similar? How does it fit into the full picture? So they're being my peer reviewer. But I am, there are questions. So I'm going to finish this sentence and go to Rita's questions. So I am, but I still am the author of the primary report. I'm the author of the data. This peer reviewer, peer review pathologist, is giving me like advice, giving me feedback, and then we have to agree on this. But the final decision, or disagree, we can disagree as well. If we disagree, too bad. We have to figure stuff out. But the final decision is mine. And that means that what peer review pathologist is doing, and currently you can read a study on glass and have a peer reviewer review it digitally. So their information is not really data, because data is my report. Only what I put in this report is data. So there is a little bit of controversy. Oh, do we really need to retain those slides if it's only for peer review? There is a little bit of discussion in the industry because we currently don't do it. But this document is open for discussion or for comments. I think maybe this period is already closed, but everybody who wanted to comment on that already had the chance to do that. And hopefully there is going to be a different version later. So, yeah. Another thing to this question too, and we have like 8 questions. I'm spending a lot of time on one question, but this basically disappears if your primary review is digital. Then you have to retain those slides anyway, and it doesn't matter if peer review was done on digital, on glass, you retain your slides. So basically, assuming at some point everybody's going to go digital and do digital evaluation, you don't have the problem anymore. You have to archive your slides anyway. So let's address a question. Question from Rita: for how long should we retain the digital image? Same as the glass slide. Yes. So this is all in the institution standard operating procedures, but basically, because this is what the data was generated from, it has to be retained as long as all the other study material. So yes, it basically becomes another piece of data in this instance because it's digital, but another piece of something that we have to keep with all the other stuff that we're archiving for a study. And let us move to question number 3. Question number 3. Just very quickly, did you know I signed up for TikTok? TikTok is scary. I don't know how many people in our community are on TikTok, but if you are on TikTok, find me under the handle Alex Żuraw Digital Path, and you spell it ALEKSZURAW Digital Path. This is my handle on TikTok, or maybe your kids have TikTok so you can see what I produce there. But now back to the guidance. If the whole slide images files are retained, should the glass slides also be retained? The answer here is yes. They quickly answered in the first sentence. So, and that kind of goes back to your question, Rita. The glass slides are study specimen and must be retained as study specimens after study finalization in accordance to this 21 CFR per 58. This is Code of Federal Regulation that we are following if we're doing GLP work. And then, question 4, what should be retained with respect to the whole slide image file? Should modified whole slide images files be retained? Yeah, image files be retained, sorry. And the question is, the answer is here. So first of all, when we're capturing those images, we should not modify the file. So then if you don't modify the file, then there is nothing additional that you are retaining. But often modification happens when you are doing image analysis, and image analysis is being done for GLP studies as well. And then in that instance, you do retain the modified files. You do retain the originals, you do retain the modified. The purpose is slightly different. It's not just primary evaluation that you use digital instead of glass, but so you have different type of outputs. You're maybe counting something or trying to quantify something with image analysis tools, and you then do retain it. But let's check verbatim what they're telling us. Yes, so here, the whole slide image files referred to here as the original whole slide image files should be retained. So the originals, we retain the originals. But any technical image processing modifications made to whole slide images files prior to being provided to pathologists should be documented and retained. This is not really happening, and you should get the original. As a pathologist, you should get the original, and so then you don't have to document anything. If there is some smoothing, color manipulation happening before you get the slides, then of course you have to document it and retain this information. But here they also say that viewing software should not allow the original whole slide image to be changed. So you are just viewing the slide. What you can do there, like you can do on your computer screen, you can adjust the brightness, adjust some contrast, but this is something you do under the microscope as well. So this is allowed, not like regulated, doesn't have to be documented, because you are not changing the original image. So this is totally fine. Here, brightness, contrast do not need to be documented or retained. It's basically my personal preference as a pathologist. I want it brighter, less bright, whatever. I can change it and not tell anybody about it. Ain't that cool? I think it's cool. So now, here another question of ours. Should written procedure for whole slide imaging process be in place? Yes. Basically, if you work in GLP, you need written procedures for anything. And the more I work in GLP, the more I realize it. Before, when I started, I didn't know how strict that is. But in GLP, there's even a format mandated for how you sign stuff, how you put in the date, how you correct input data. Like, you have written procedures for everything. So obviously, this is a big deal that you can now look at slides instead of under the microscope on the monitor. So yeah, there should be written procedure for that. So yes, that's what FDA says. FDA says yes. Should be in place, written procedures. They might include scanning, validation, training, maintenance, software version control, backup, disaster recovery, everything. But this everything, do not be terrified. This everything is what you usually do for any computerized system and for anything in GLP, good laboratory practice. You have to protect from all this stuff, from virus protection even, and disaster protection, anything. So yes, the answer is yes. Question number 6. Should the whole slide image systems be validated? I'm actually surprised that there's no yes here, but there is a qualifier. So again, if we are using whole slide images instead of glass, which is in lieu of the original glass slides during histopathology assessment, and here this peer review as well. Well, peer review, yeah, that's fine. The archival part was a little bit controversial. Performed under GLP, then yes, we have to validate. Is that a surprise? No, because we have to validate everything for GLP. If you are doing non-GLP, then you don't have to validate. But for GLP, yes, any computerized system. So why am I saying a computerized system here? Because digital pathology system is a computerized system. So basically, this this guidance that we're going through is kind of telling us how to deal with whole slide images as a computerized system, and how to apply whole slide images to what is already written in the 21 CFR Part 58. Part 11 is for computerized systems. If anybody knows exactly, I can Google it later, but I think Part 11 is for computerized system, Part 58 is for the non-clinical work. But there is a GLP regulation for computerized system, and there's exactly written what do you do with a computerized system. So you take this digital pathology system, you say, oh, it's a computerized system. What are the components of the computerized system? It's this, that, scanner, viewer, whatever, uploading of slides, and you validate them. So yeah, no surprise there. So going to our next, here, FDA themselves says should be validated and maintained in manner specific to the intended use. That's important as well. Intended use. Oh, can I change? Yeah, intended use. So intended use, which is what you specify in your SOPs, and basically it's specified a system should not be used in a way that's not intended to be used. So important thing. But basically, yes. Okay, let's go back to yellow. And question number 7. How should whole slide image files be protected, including when transmitted to external users? Well, so these are digital files. We have to protect them somehow. So if they are, again, assessed in lieu of the original glass slides, because also at the beginning of this document, it's written there that it does not apply to pathology consultations and education and like all those ways that we already are using whole slide images for everything else than primary study evaluation. So for that, we can use them, no problem. But here, if we want to use them again in lieu of original glass slides, we have some stuff to take care of. So generation of backup file, chain of custody, access controls, securing data systems and data transmission, written procedures again, in compliance with electronic records. So this is becoming an electronic record. And in any, if at any point of time you have a question, give me the question. I'm happy to answer the question, hopefully, or refer you to whoever knows the answer to this question. So, but basically, this slide becomes then electronic record. So digital pathology system is a computerized system. When in doubt, you go to, oh, what do I do with a computerized system? A digital pathology slide is an electronic record. So when in doubt, how do you deal with electronic records? And this is basically what you do with electronic records. You have a backup file, you like have a chain of custody, not everybody can access, and everything that you have to do to be compliant. So that is taking us to our last question. And the last question is, should the signed pathology report / peer review statement state that whole slide images were evaluated in lieu of glass slides? Without looking at the answer. The answer, of course, is yes. Yes. Everything has to be stated, everything has to be clear, everything has to be traceable. So yes, we have to say this particular study was evaluated on digital slides. If we're doing GLP, if we're doing primary evaluation for GLP on digital slides. So again, yes, we have some yeses. Good. Fantastic. Yeah. Also, if pathology peer review is conducted on digital slides, then we say that it was done on digital slides, given that we have validated everything, systems work, everything, there are SOPs, standard operating procedures in place, and all that jazz. So this is what FDA is telling us about using of whole slide images. And why I'm so happy about this document as well is because here they are saying do what you should do for computerized systems, do what you should do for electronic records. We kind of had that information already before, because it's not the first computerized system, and it's not the first electronic record that is being retained or dealt with under GLP. But now FDA is actually telling our community, our digital pathology community, hey guys, here it's guidance. It's just draft guidance, but we know you are using it. This is how we are fine with you using it. This is the way you can apply your current regulation to this new form of data, new form of computerized systems. So thank you very much, FDA, for putting out that guidance. Even though it's draft, even though we are still waiting for comments, there is a guidance for the industry. So for anybody who is questioning or who is asking themselves, can we use those whole slide images? Like, how do we use them? It's written right here. Thanks so much for listening, not just today, but throughout the year, for following me and for wanting to advance digital pathology, for wanting to do it in your workplace, for wanting to overcome all the obstacles, and for just being a digital pathology trailblazer. And if you know somebody who will benefit from this episode, who is working in a GLP environment and they're looking into this, if you could share this episode with them, the best would be the link to the Digital Pathology Place website, because on the website I will have the audio episode, the show notes, the transcript, and the video, the live video that I had about the guideline. So if you can share this with them, that's going to be a fantastic Christmas present for me. Thank you so much, and I talk to you in the next episode.